Hello and welcome. Thanks for joining us here for our weekly seminar series hosted by the Living Earth Collaborative and Washington University's Ecology, Evolution, and Population Biology group. Today, I have the great pleasure to introduce our speaker, Dr. Don Miles. Dr. Miles holds many hats, but I think it's fair to say that he would consider himself an evolutionary ecologist, if nothing else. And he's currently the Clark Way Harrison Distinguished Visiting Professor here at Washington University. And I'll speak on behalf of everyone over in our wing of McDonald Hall that we're very fortunate to have him on loan this semester from his position as a professor at Ohio University in Athens, Ohio. Dr. Miles' academic career started at the University of Pennsylvania, where he worked with St. Louis's own Dr. Bob Ricklepps. And before joining us here at WashU, Dr. Miles has held a genuinely remarkable list of prestigious appointments at institutions all over the world, including at CNRS, the University of California, James Cook University, and the University of Washington. Along the way, he's been funded by the National Science Foundation, National Geographic Society, and the USDA. This work has led to nearly 100 peer-reviewed papers and book chapters, including in prestigious journals such as Science, Nature, Proceedings of the Royal Society of London, and the American Naturalist. I'll also mention that his 2010 science article is perhaps the paper in this field that I personally admire the most. So it's a real thrill to have Don here. To finish up the flattery stage of the seminar introduction, I'll be honest that it is quite hard to synthesize down all of the extremely insightful contributions that Dr. Miles has made to ecology, evolution, physiology, and behavior. But I think perhaps the best way to convey his impact on the many different fields in which he works is simply to note that when students ask me about almost any topic, his articles disproportionately populate the list of readings that I give to them. So one last thing before I turn it over to Dr. Miles for what's sure to be a really terrific seminar, I'm going to remind all of you in the audience to please leave any questions or comments that you have in the chat section next to the video screen that you see. And we will relay them to Dr. Miles at the end of his presentation. And so with that, I'm going to stop talking and I'm going to let Dr. Miles take it away.
Great. Thank you so much for that warm introduction, Mike. Thank you to Matthew Austin for asking me to present the seminar. Thank you to the LEC Fellows for being so hospitable. And to all the grad students and faculty I've met, it's just been a delight to be here. I really feel lucky to have this semester working at WashU. So what I'm going to do today is to discuss a bit about my research that looks at physiological and behavioral strategies to cope with rapid warming, and I'll focus on lizards, although I have worked on a variety of other taxa. I first want to begin by acknowledging the various institutions with whom I have worked and who have supported my research. And in particular, I want to thank my colleagues who have collaborated on the work with the tree lizard, Fausto Mendez, Carla Piantoni, Jim Brown, Howard Snell, and Anthony Gilbert. In fact, I'll be presenting much of Anthony's dissertation work today that we worked through together. And with the Zootoca work, the common lizard work, I'll present Jean Claubert, Lara Zamorano, Sandrine, Andreas, and Alexis Ruchman were instrumental in much of the work that we did. And I also want to pay my respects to Phil Rosen and Barry Sinervo, both of whom have died in the past year. And I also don't want to neglect mentioning the number of undergrads whose contribution to the fieldwork made much of this research possible. So most of my work, I would say, involves asking how environmental variation affects the ability of organisms to persist in environments that may be cyclical in temperature variations, cyclical in rainfall, et cetera. And we can look at environmental variation in a diversity of ways. We can look at it in a diurnal manner, seasonal, comparing winter, spring, summer, and autumn seasons, annual cycles, decadal cycles like El Nino, La Nina, or the North Atlantic Oscillation. But in general, these phenomena have a periodical component to them. They're predictable in some sense, and what may vary in the terms of these decadal oscillations is the intensity with which these cycles are exhibited. Yet now we are facing a situation in which we are experiencing unprecedented warming. This is a snapshot from the Berkeley Earth Organization taken in June. And what you can see is the temperature anomalies globally. And the thing to note is the massive amount of red in the United States, Africa, Europe, Asia, and Antarctica. We are experiencing temperatures that have not been seen in millennia. Much of this is driven by the increase in atmospheric CO₂. This is a recent plot of the data from Mauna Loa volcano, looking at the CO₂ concentrations. We have broken the 400 parts per million layer. We are now resting somewhere between 415 and 420 parts per million CO₂ in the atmosphere. And the concern is that our atmospheric concentrations have not been seen since 20 million years ago. This is a plot showing past estimates of CO₂ going from different time periods from 1000 to 2000 and extrapolated to 2500 based on global circulation models, data from the Antarctic ice core in thousands of years, and then we move into millions of years. And this arrow portrays where we are in the scale of CO₂ in relation to past atmospheric estimates. What does this mean? Well, if we look back in time, what we are seeing is levels of CO₂ that have not been experienced since the early Miocene and late Oligocene. This means that we are likely to see climates that have not been on Earth or experienced by Earth in millions of years. And based on current trajectories of CO₂ production, we are likely to approach Eocene-like temperatures. Now, if we look at temperature anomalies going from the 1800s to 2021, what we see is that we are about 1 degree C above the normal temperature as recorded by the baseline data from basically 1940 to 1960. So what this means is we're seeing unprecedented kinds of temperatures. Records are being broken by the day during the summer. In 2021, Sicily recorded the highest temperature ever recorded in Europe at 48.8 degrees C. And the reports from NOAA show that 2021, the July temperatures were the hottest month ever recorded based on 100 plus years of records. Not only is temperature getting warmer, but we're seeing shifts in the amount of precipitation falling in different parts of the globe. And what one can see here, this is a plot showing the change in annual precipitation comparing 1984 to 2016 to the mid 20th century. And you see there's a lot of brown here, indicating a decline of 4 inches on average. The Western United States are drying up, and notably, Southern France and Northern Italy are also getting drier. They're experiencing substantial drought episodes, which is unusual for that part of the world. Not only that, but if we look at the US, the western part of the US is getting drier and the eastern US is getting wetter. So we're seeing a redistribution of precipitation, which has an impact on net primary productivity and consequently the amount of resources that are available for organisms that depend on insect productivity for their daily energy requirements. Now, this climate change is producing novel kinds of weather patterns that species on Earth at the moment have not experienced. Now, this slide looks like bushes with the leaves on the ground. Those aren't leaves. Those are budgies. Those are dead birds that succumb to the high temperatures experienced by Australia in the past few years, where temperatures exceeded 50 degrees C in some parts. So what we're seeing is species being exposed to novel environments in which the rate of change is likely to exceed their potential adaptive capacity. So responses to climate change, and I'm sure many of you have seen a figure like this before. We have species tracking their niche by shifts in their distribution, so they move with their favorite habitat or their preferred habitat. There's an adaptive response, either through evolutionary change and traits that allow the species to persist, or plastic responses. And in the end, that species cannot keep up with the changing climate. There's the failure to adapt, which leads to extinction. Now, this extinction pattern is not going to be a wholesale extinction, but the persistent drip of local populations going extinct until you have global extinction. And we're seeing that now, at least in lizards and other terrestrial taxa. So what are the responses to climate change that we can see? Well, since current predictions suggest rising extinctions across species, and the majority of studies focus on estimating extinction risk or probability of persistence, how can we get into the important topic of what are the traits that will allow species to survive in an altered thermal environment? And so what we need is information on interspecific variation in thermal traits, which we have surprisingly little amounts of data on that. And in particular, fewer analyses focus on local population dynamics and functional traits in order to make predictions about how differences in these thermal traits can facilitate a population coping with these novel thermal niches. So some of the biological consequences of changes in the abiotic factors include altered habitat characteristics, loss of shade from drought stress, for example, as plants become water stressed, changes in thermal opportunities, which limit the ability of organisms to find suitable thermal microhabitats, a potential higher risk of dehydration, ultimately leading to a reduction in food availability. And because of the high temperatures and the rising body temperatures, higher metabolic costs. And Huey and Kingsolver called this metabolic meltdown. In their idea, what we have is a population who shows a distribution of body temperatures as shown in this lower panel. And as the climate changes, there's a compensatory increase in TB as organisms are coping with rising temperatures in their environment. Now, if we look at this upper panel, this portrays the trace of net energy gain. And what we can see is that we have areas of positive energy gain, which leads to growth. And then when you have the curve crossing this line, which would be zero energy gain, you have a negative energy or negative growth balance. So what we're seeing is TB increasing. What's the response of organisms to that increase in body temperature? Well, one prediction is that we should see a change in this energy gain curve such that organisms shift their thermal preference and their thermal optima to lower values to avoid the potential for reaching this part of the curve where they're actually losing energy and losing weight, which has a strong negative selective consequence. So warming is going to lead to metabolic meltdown if it's combined with a decline in food availability. So in terms of metabolic meltdown, the predictions are that food-stressed lizards, or you can insert any taxon here, should have a reduced thermal preference. They should invest in energy conservation behaviors, which means they would lower investment in locomotion, since that's a very expensive activity. They would shift their optimal temperatures for performance to lower temperatures so that they can maximize their temperature or their thermal sensitivity to whatever task it is by being more efficient earlier in the day where temperatures are more moderate. And then there's also the potential for heat hardening, where organisms become acclimated to the higher temperatures, and therefore they have a short-term response to this. Now, there's also an adaptive response where you see selection on thermal traits and local adaptation to thermal traits. So what I want to do is to look at two systems and to look at two stories. One is a North American desert lizard species, and this species has had a history of adaptation to arid environments. And the species has been exposed to chronic prolonged heat waves and drought over the course of its history in the arid climates of North America. In contrast, I want to present some data on a European mesic-adapted species, a cool climate lizard species, that is now experiencing episodes of drought and heat waves that have not been part of its natural history, and how that change in climate is leading to population declines and ultimately extirpation of local populations. So the first thing I want to look at is how are the thermal traits that are critical for persistence in an environment and physiological performance affected by one consequence of metabolic meltdown, and that is food deprivation. So the focal species is the tree lizard, Urosaurus ornatus. It's a small-bodied lizard. It has a cohort generation time of about 1.2 to 1.5 years. Most individuals mature by the beginning of their first year. Hatchlings appear in July, and they reach sexual maturity by April of the next year, April or May. Some lizards, some individuals can live long. I've captured some individuals that were at least 6 years old. But this lizard is an arboreal species. It's insectivorous, and consequently, it makes an ideal species to look at how a change in food availability may affect its performance capacities. The reason being, it relies on food on the tree. And if the trees are drought stressed and there's low food availability, it has both the consequence of higher substrate temperatures and lower food availability. So what we did, Anthony went out to Arizona, and he measured baseline thermal and performance traits. He measured thermal preference, which is the temperature selected by an organism in a thermal gradient without cost. So it's the temperature that it would select in the absence of predators, etc. We measured endurance capacity and the thermal sensitivity to sprint performance. We then fasted lizards for 13 days. And then after that, we measured their same traits after 13 days. And the mass loss we recorded after 13 days, males lost about 9% of the mass and females lost about 8% of the mass. And I would like to mention that the females were not gravid, so we did not include gravid females in the experiment. This is just to show you what a photothermal gradient looks like. It is warm at the upper end, as you can see with the lamps, and then there are ice packs at the lower end, which keep it cool. And so we are able to generate a thermal gradient of about 50 degrees C to 18 to 20 degrees C. Here's a racetrack. We sprint the lizards down the track. There are lights along the length of the track, and as the lizard occludes the beam, there's a computer that records the temperature, and then from that we can estimate velocity. And I don't have a picture of the treadmill. So what did we find? All right, well, after 13 days, we ran the lizards on in the thermal gradient. And prior to the food restriction, the preferred body temperature for the fed and fasted lizards were indistinguishable. So there's no difference in the preferred temperature selected by the lizards in that thermal gradient. After the 13 days, we see a marked decline in the temperatures selected by the fasted lizards. No difference in the temperatures selected by the fed lizards. If we look at the endurance, we have endurance in seconds on the y-axis, We have before and after, and we have lizards that were run on the treadmill at 36 degrees C, and then again at 29 degrees C. We wanted to compare the two temperatures, one in which the lizards were at a lower temperature, which would be likely to occur if they were selecting cooler microclimates, and then the endurance that a lizard would display when run at their field active body temperature. Now we have males in the black lines and females in the lower line in the open squares. And what you can see is that a consequence of fasting is a decline in endurance in both males and females. It wasn't significant for the females, but it was significant for the males. And in fact, the endurance was about 20 seconds on the treadmill. And now I would mention that the treadmill was run at a belt speed of half a kilometer per minute. So this would involve the lizard not having the capacity to run a few 100 meters. If you look here at the 29 degree endurance, same sort of pattern. Males and females are showing lower levels of endurance as a consequence of fasting, and the slopes were similar. So we see that one consequence of fasting is loss of endurance or reduction in endurance, presumably representing an energy conservation strategy, since endurance is a very expensive energetic activity. Now, we also looked at sprint speed, and what we are portraying here is the results of a generalized additive mixed model to create these thermal performance curves, these thermal sensitivity curves. And we have the temperatures that the lizards were run here. The curves are anchored at the critical thermal temperatures, so CT min at about 14 degrees C, CT max at 44.5 degrees. And then we have mass here. So we included mass as a covariate. So we have light lizards here, heavier lizards here. And then we have endurance on the y-axis. And what is evident in the fed lizards is the performance breadth, which is the temperature at which the lizard achieves 80% of its maximum performance. It's narrower for the fed lizards. And if you take the maximum endurance value and go down here, you find that the optimal temperature for performance is about 36 degrees. Conversely, if one looks at the fasted lizards, we see a shift in the critical thermal limits. The fasted lizards have a CT min of about 13, 12.5 to 13, and slightly higher than the fed lizards, but not significantly so. But what one can see is that first, the performance breadth is broader. So they are doing 80% of maximum across a wider range of temperatures. But not only that, the thermal optimum has shifted downward so that their optimal temperature for performance is now about 34 degrees C. So we are showing that the predictions of the metabolic meltdown hypothesis is confirmed by looking at the shift in performance over a variety of temperatures. What we also did, I'm not showing, the data for sprint speed show the same thing, so I'm not going to show that in the interest of time. What we did is we asked, is there any evidence that heat hardening might be a potential response? Now, these lizards are in an environment where they can experience short-term, extremely high temperatures. So the temperatures, air temperatures at least, can go up to 40 to 43 degrees. Substrate temperatures can be even higher, maybe 50 to 55 degrees. It's a transient increase, but we were interested in knowing how do these lizards cope with the short-term thermal stress. And this would be relevant for species that are exposed to heat waves, or in France, the canicule. In this case, ambient temperatures would approach or exceed their critical thermal maximum, or the temperature at which lizards lose the ability to perform coordinated activities. So they basically lose their ability to locomote. They lose their ability to basically behave. And you can think of this as being an ecological critical point where they're effectively ecologically dead. And so we asked if lizards are exposed to a short-term pulse, an acute pulse of heat, what are the phenotypic traits that are modified by that hardening response? So what we did is we looked at two populations, a low elevation population along the Rio River in Tucson, and then we looked at a mid-elevation population at the Appleton-Whittle Research Ranch outside Elgin, Arizona, which is a semi-arid grassland. And if lizards were going to shift their distribution, the semi-arid grassland would be presumably a refuge for desert-adapted lizards. So what we did is we measured CT max twice. That is, we took lizards to their limit before reaching a lethal temperature. We did that by heating them under a lamp and following their behavior until they lost the ability to turn over when we put them on their back. And this is called the loss of their righting response. So we did that twice. And then what we did is we evaluated the change in their CT max, their thermal preference, and sprint speed. And so this is just to give you an idea about what the habitats look like. This is the semi-arid oak savanna at the Appleton-Whittle Research Ranch. It's a beautiful location, scattered oak trees, and the lizards are found on these oak trees. You rarely see them on the grassy slopes, largely because of the presence of a suite of snake predators that will easily consume many lizards. And then here on the Rio River, this is a mesquite bosque, a mesquite woodland. The trees have shade, but when the trees are drought stressed, they do drop their leaves and the thermal climate increases. So the benefits of the shade afforded by the leafy petioles are no longer there. So essentially what we're saying is, given a heat pulse, these mesquite trees would lose their leaves, and that would expose the lizards to a pulse of warm temperatures. So what we did is we measured the peak response to the heat shock. And so we measured the lizard's response at 2, 4, 6, 8, up to 24 hours. And what we found is that the heat shock response was transient. It peaked at 6 hours. There was still a persistence of some response, but basically when we looked at the change in CT max, it was maximized at 6 hours post heat shock. If we look at what happens to the lizards after the heat shock treatment, the pink line here represents the thermal preference of the lizard prior to the heat shock treatment. The purple line represents the treatment, or sorry, the T-pref after the treatment. And you can see that there is a drop in T-pref of about 4 and 4.5 degrees. This is for the lizards in the oak savanna. This is for lizards in the desert. This top panel represents the body temperatures that we reported at the oak and desert habitats. And so the lizards are characterized by warmer body temperatures in the desert than in the oak, which is not surprising. It's a warmer environment. The TE distribution, and TE, I might mention, is the the temperature attained by an organism in the absence of any thermoregulatory compensation. So it's the sum of thermal inputs, so it's the sum of heat in minus heat out through convection and conduction and re-radiation. So there's a negligible component to heat production by metabolism or heat loss by evaporative cooling. And so what you can see is that it's much warmer in the desert environment than in the oak environment. And so lizards, by shifting to a lower T-pref, have to go to a cooler microclimate. So they are going to be spending more time in the shade than they would be in the open. This lower panel is the TE values for sunny exposed perches. And again, most of the perches are well above the CT max of the species. And so lizards would have very limited time in which they could be in the sun post heat hardening. They were doing a little bit better in the oak savanna, largely because the the TE values are more moderate. All right, so what happens with sprint speed? Well, if you look at the sprint values, we took the lizards and we immediately ran them after CT max. So this line, the dashed line, represents re-eto. The solid line is the oak savanna. And just after the heat shock, the two sites differed in their response. Both, however, declined in velocity, and that persisted 4 hours post-heat shock treatment, and then begins to recover at about 6 hours. Notably, both populations showed an increase in CT max after the initial exposure to CT max. So what they were able to do is have the ability to tolerate even warmer temperatures as a consequence of exposure to that initial heat shock. Now, what we see here is that there is a cost of heat hardening with a decrease in sprint performance. We did the same thing for populations at different elevations. We have here the change in CT max as a consequence of the initial exposure to high temperatures along an elevational gradient, going from about 600 meters to a little under 2,500 meters. And what you see is that populations at the higher elevations are characterized by having a higher heat shock response than the populations at the lower environments. Oops. And we hypothesized that the heat shock response was probably greater because these lizards are rarely, if ever, exposed to such high temperatures. And so they've have a more naive response to high temperatures. In contrast, populations at lower elevations are probably already running at close to CT max. And so the heat shock response is not as great. And then if you look at the change in CT max before and after, so this is the initial shock, this is the post-shock, what we see is that lizards at the low elevations tend to have higher values of CT max, the lower and lower response, and then the species, or sorry, the populations at the high elevation have the highest response. Interestingly, we have the 850 meter population clustering down here and the 1400 meter up here. So there was a bit of a mystery about why that happened. And we think that what's going on is this is a more open savanna, and this is more of a closed environment. There's more shade. All right, the last thing about your source is there an adaptive response? And so what we were interested in is there any selection on thermal traits? Because if there is a shift, if the traits aren't open to selection, then their ability to cope with new thermal environments may be compromised. And so what we did is we asked, is speed, CT limits, and T-pref favored by selection? And so we collected or captured 106 females and 100 males. We performed, this is using a capture, mark, recapture approach. So we captured the individuals, marked them, and then performed the measurements, and then released them, and then recaptured them. This is just to show you the thermal environment that the lizards experience. The dashed line here is their T-pref. The CT max is their, I'm sorry, the red line is their CT max. And you can see this value here is the TE max. And so there are periods of time where the environment is too warm for them to be active. But there's a considerable amount of time where they can be active, where the temperatures are within their preferred temperature range. And so that would be the difference between the CT min and TE min and TE max. So when we looked at selection, we got these selection profiles. This is for survival for against thermal preference, survival with sprint speed measured at 36 degrees, survival at performance measured at 40 degrees, and then survival against CT min. And what we see is that there seems to be a directional selection for survival, or for survival with thermal preference. And the same is true for performance at 36 degrees. And no evidence of selection for performance at a higher temperature, which was surprising. And then we see survival going down as CT min goes up. And this is summarized in this next table, where we see significant selection values for T-pref, P36, and P40. Now, these are selection differentials. They're not the selection gradients. And the only two that come out as being significant are T-pref and P36. And the reason is that T-pref and P36 are coincident. Now, we looked at correlational selection, and what we found is the only significant correlation was between that predicted survival was a negative correlation between CT max and T-pref. So basically, lizards that had warm thermal preferences and low CT max values were favored by selection. Now, this is a bit of a conundrum, but we think what's happening is the lizards that are showing high thermal preferences are selecting temperatures or microhabitats where they are not exposed to high temperatures. And we hypothesize that this might be an example of counter gradient selection operating on these two traits. All right, now I want to switch gears and look at the European common lizard, Zootoca vivipara, probably one of the most studied lizards on the planet. And this is the beast. It's a small-bodied lizard. It matures in 2 years. Populations can be either oviparous or viviparous, which is unusual for species. I contend they're separate species, but there are systematists who disagree with me. They prefer boggy habitats, so they're mesic adapted. They have the broadest distribution of any terrestrial vertebrate. So they occur from Spain, England, through Scandinavia, all the way through Europe and into through Asia and into the extreme eastern part of Asia. They have isolated populations in Slovenia and Croatia, and they have this interesting disjunct population here. So this is vivipara in the Pyrenees, which are oviparous. There's a gap here, which is probably related to the lack of suitable habitat. It's not very boggy. And then you have this distribution of the viviparous forms throughout the remainder of the range. Now, this is this typical habitat for Zootoca. And working with desert lizards, it was a little bit difficult to get my head wrapped around working on a lizard that lives in water. And so I had to educate myself on how to be a lizard. But you see, there's a lot of grass, scattered trees, and so they prefer these boggy environments. And the lizards will bask on the rocks, or they'll climb up on heather, or they'll get up in the pine trees. And the best thing about this is catching lizards, because you don't use a pole or anything, you just catch them with your hands. And so you'll see people walking in the field, and all of a sudden it looks like they're having a spasm. They fall to the ground, and then they pick up a lizard. So it's a great way of spending the day. And so this part of the talk is based on a study by Elvire Bestion and her co-authors, in which they reared zootoca in a metatron, and they exposed the lizards to a warm climate, and that was basically 2 degrees above ambient temperatures, and then the present climate. And they followed the lizards over time. They had females gave birth. They measured the juveniles, and what they were able to show is that in the warmer environments, zootoca juveniles exhibit higher body growth, so high that they are able to reproduce their first year, and in the wild they reproduce in their second year. So about almost 70% of the neonates, those born the first year, are reproducing the very next year. That comes at a cost. And the cost is that those lizards born in a warm environment suffer higher mortality. So their annual probability of survival declines. So what we did is we had this massive global change study in which we were surveying 22 populations in the south central portion of France in the Massif Central range. This was initiated by Barry and Jean in 2003. And we have been surveying the populations from 2004 to 2019, and then COVID happened, and that ended that for the time being. And in the course of our work, we identified multiple local extirpations and population declines. And so if you want to see what the population orientation looks like, here's France. This is the area in south central France where we're working, and this is the Massif Central. And this map is color coded according to the Enberger pluviothermic quotient. It's basically a net transpiration rate. So sites that are blue are receiving more rain than sites that are red or pink, and those sites are also warmer. So what we did is we were looking at the temperature data, and it was apparent to us that the increase in temperature, mean temperature, wasn't from an increase in daily temperature. It was nocturnal temperatures that was increasing. So we said, let's redo Bestion's study, but we're going to purposefully raise only nocturnal temperatures. The other thing that we wanted to know was, given that we have seen declines of different populations, for example, going back to this map, they're not presented here, but there are 2 populations down at the bottom of this map, Caroux and Montelay. And Caroux is in a drastic decline, and Montelay, that population went extinct. So what we did is we manipulated nocturnal temperatures. We captured 4 lizards from 4 populations from different elevations. We kept them in the lab for 5 weeks, and then we measured growth rate, skin color, thermoregulatory behavior, oxidative stress, and parasite load. And just to keep things brief, we found no difference in thermoregulatory behavior and oxidative stress. So that was not a response. Just to show you the background, this is the distribution of nocturnal temperatures at the 4 sites, Barnesac, Braque de Bouvier, Col du Pendu, and Vialis. And then we have here the maximum nocturnal temperatures. And so it turns out that Barnesac it's the highest elevation population. It's also one of the warmest. Who knew? And then Col du Pendu is the coldest, and then Vialis is intermediate. But this is our treatment. This is a trace of the temperatures experienced by the control hatchlings. And what we did is we took yearlings and kept them in these individual boxes. And the experimental treatment, we had the temperature at night at about 4 degrees C above the control. So what did we see? Well, the first thing we see, we looked at changes in snout vent length, change in mass, change in body condition, and change in the tail volume. And the tail volume was measured because that's a storage organ for the lizards. There was no difference in the mass of the lizards. The yearlings increased their snout vent length, which is consistent with Bestien's work, but the yearlings were in poor body condition. They were longer, but had less mass. And then if you look at the tail volume, again, a storage organ, they had lower tail volumes, which indicates that the lizards were allocating energy to growth and not storage. And consequently, it could lead to lower survival. We looked at their parasite infections. There was no difference in endoparasites, but there was a significant difference between the control and the warm lizards and the parasite infestation. So warmer lizards at night, presumably because the inability to regulate their body temperature probably ramps up their metabolic rate and puts them in lower condition for mounting a response to the ectoparasites. In terms of coloration, I didn't include the slide, but basically the lizards in the warmer environment, they had a change in luminosity and brilliance, two measures of color, such that they were able to gain heat more rapidly. Presumably, we didn't measure that, but they should be able to gain heat more rapidly than the control. Last, I want to discuss some markers of population decline. And we were interested in knowing whether there was some measure that we could use to sort of presage the dynamics of the population. Now, we have been following these populations for a bit more than 15 years. So we have data on population trends, but that doesn't necessarily tell you the whole story. So we went out one year and we collected blood from lizards, yearlings, to get a measurement of plasma osmolality, which measures water stress. And we also used the blood to measure telomere length, which telomeres are the end caps of chromosomes that over time will shorten with each successive cell division. And the length of the telomeres have been shown to dramatically decrease in individuals that have been exposed to high levels of stress. Like it could be elevated glucocorticoids or some other intermediate that leads to the reduction in telomere length. So what do we find? Well, we looked at the plasma osmolality from about 10 sites. This is a compendium of all the data. And what we found is that as you look at the mean ambient temperature, so what we did is we put out data loggers so we knew what the temperature was at each site. And we have a measurement of osmolality, which is basically how dense is the plasma, how much solutes are in there. And we noticed that for the populations that were in the warmer sites, they had higher levels of plasma osmolality, which suggests that they were water stressed. Now, if we go to those same sites and we look at body size of the individuals, these are body size of the yearlings, sorry, we measured the minimum temperature. So this is the temperature at night. And we looked at the body size of lizards from different populations. Each of these squares represents a different population. I'll show you the populations in the next couple of slides. But basically, lizards that are in these cool environments have smaller body sizes, and that's snout vent length, compared to the lizards that are found in the warmer environments. In addition, based on our abundance data, lizards from the warmer sites were showing a greater change in abundance than lizards that were in the sites that were cooler. So this suggests that there is a cost of warmer nocturnal temperatures in terms of population dynamics. So these are the populations. We could go back to the map to refer to them, but basically we had these populations. These are the populations that were declining. These are the populations that are holding steady. Now, these populations differ in elevation. They differ in mean temperature. And so they have the, and minimum temperature as well. So you look at this, and this is abundance measured as females captured per hour per person per hectare. And then if you look at female abundance in a different way, in terms of population abundance, so lizards captured per person per hectare, the important thing here is that the female abundance is a reliable indicator of the population overall. All right, so we know that there's some populations declining, some are stable. What do the telomeres tell us? Well, what we found is that telomere length is lower in collapsing populations. So just to refresh you, we have this population Puy-Marie. This is a population Montelay, and then here's Caroux. These are all showing a downward trajectory. In fact, we were sampling Montelay, and one year we caught only female lizards. There was no recruitment the previous year. So what we see is, this is a PC score, and it's a composite score of mean temperature, minimum temperature, and elevation. And so for the populations that are, in abundance, and the populations that are declining, they have a significantly lower telomere length than populations that are either not threatened or growing. And this is corroborated by Caroux, which recent surveys shown that we only found males, so that we couldn't find any females. Notably, these populations, at the time that offspring are being born, they're drying out. So normally these bogs would be wet in late July, early August, when the females are giving birth. They're not. And so hatchlings, neonates, are released into the environment in a very thermally as well as hydrically stressful environment. All right, so what we can take away from this? Well, there's evidence of plasticity, but it's not necessarily positive. There is costs to the plasticity. We do see that there's some local adaptation evident in thermal traits, as evident in the comparison of heat hardening across elevations. There's evidence of selection, so that there's the possibility for selection to move the traits in the direction that would facilitate persistence in the environment. Notably, nocturnal warming is probably the important factor that's influencing phenotypic traits. And higher growth rates are happening at the expense of body condition and potentially future survival. And then lastly, we can use telomere length to identify populations in decline. And with that, I will end, and I am happy to take questions. Thank you so much for listening.
All right, thank you, Don. I'm sure that everyone at home is giving you a round of thunderous applause for a very excellent
I can hear it from
seminar.
here.
Yeah, it's so loud it reverberates all throughout the city of St. Louis.
Stunning.
So you've already gotten some questions in, and I'm sure that some more will roll in as we go through the question and answer period. But so I'll start off with one from Caleb Axelrod. And Caleb notes that he thinks that your marker capture selection study is really interesting, and he's interested in particular in how selection acts on sort of the thermal plasticity. And so he wants to know if you've thought about doing kind of similar selection experiments for kind of trait flexibility, so like the change in an individual's Tmax or the decline in their performance, as well as on the traits themselves.
Yeah, that's a good observation. We actually have the data because we do have the change in CT max from the Rieto population, or not the Rieto, but the Appleton-Whittle population. So, I mean, that's something we could do. I think to really get at it, we'd have to have larger sample sizes to really nail it. Because what you'd have to do is a multivariate selection analysis. And so you need some substantial numbers in your sample. You know, one thing we found is that there wasn't much difference between the sexes in terms of the response. You know, females were lower, but the trend was more or less the same. And that could be a consequence of differences in body size. So we didn't, for endurance, we didn't correct for body size. But that's, I mean, that's, that would be a worthwhile project to pursue in the future.
Big undertaking, though, I'm sure.
I'm going to do it next field.
Yeah.
Thanks, Donald. That was a terrific seminar. Thank you. That was really fascinating. We have another question from Adam Smith. Thanks, Adam, for watching us. He says, first, fascinating. The thermal responses in the treadmill experiment look asymmetric. And here comes the question. Do you think moving toward warmer climes like we are now would yield similar or different results from moving towards cooler climes, for example, glaciation? And also says, ergo, would starvation under a cooling climate make lizards prefer warmer climes?
Ooh, that's an interesting question. Yeah, you know, I would imagine that if the circumstances changed, that lizards in a cooler climate would probably not show as dramatic a decrease. That is, they're already in an environment that may be less optimal thermally, as too cold. So they wouldn't necessarily show as great a shift in T-pref because there isn't a cost. They're actually, the cost is being too cool. So yeah, that'd be interesting to investigate because there are instances where you know, for example, the desert environments can experience less warm temperatures. So one thing that's happening is an increase in cloud cover, which can lower temperatures. So lizards are experiencing cooler temperatures than normal. So that would be an interesting thing to investigate. Yeah.
So I have a question that I would love to hear you talk about. So I'm interested a lot in this metabolic meltdown idea, and for your lizards in sort of these really arid climates, I'm interested. You know, part of how climate change is going to sort of change the climatic environment. Obviously, is through warmer air temperatures, but we also expect in some regions that there's going to be a lot less precipitation, all these other things. And we know often that temperature and sort of temperature and desiccation kind of interact in important ways to influence sort of organismal performance. And so I guess I'm curious how you think the predictions of the metabolic meltdown hypothesis might change if you also incorporate kind of changes in precipitation or changes in sort of water availability.
Yeah, so you know there is an increased interest in not just thermal regulatory strategies, but hydrothermal regulatory strategies. And if you go into the frog literature, there's several papers where the frogs are both run, some measure of performance is done at different temperatures, but then it's also done at different levels of water content. So the frogs are allowed to desiccate to 10%, 15%, 20% of their body mass. And what they're finding is that the thermal performance curve for temperature is radically altered as a consequence of the inclusion of hydric stress. And for a lot of a number of years, it was thought that you don't have to worry about water stress in lizards because they have impermeable skin, blah blah blah. And it turns out that with the data that we're getting with plasma osmolality and also water loss data, lizards are leaky. And so I think what one has to do is incorporate both hydric condition and temperature. And then, so my expectation would be that in terms of how that, how that energy gain curve shifts with water reduction, I think it would really be pushed further over. because water is so critical for maintaining mass balance equilibria. So you could, I think using some biophysical first principles, you could make a prediction about how that curve should shift with the synergistic action of temperature or rather lower food availability and lower water availability.
Do you think that it would more negatively, do you think it would have a bigger change on your arid lizards or on sort of the lizards that are used to having more water? In other words, are the arid lizards going to be closer to the limit, or are they a little bit hardier to water stress?
I think it depends on having data showing how leaky the skin is. So, for example, in the common lizard, we found that lizards that were in drier environments, they actually had leaky skin, leakier than we thought. We expected, much lower rates of water loss. So I think that's something that one would have to first characterize to demonstrate that arid lizards are less leaky, and then to make a prediction about how the leakiness changes with temperature. Now I have some data that I collected last summer, where I have this device called a vapometer, and it measures the resistance of water loss across the skin. So you get instantaneous measurements. And one of the things I want to do here is to see for the populations that I've measured. So I measured lizards from extreme arid environments up to more mesic environments. And so I have the data to answer that question. So I think it's something that we haven't really looked at because we just thought lizards were airtight or watertight.
It's really fascinating. Thanks.
Thanks, Don. Here's another question from John Birmingham. He says, how do the lizards get rid of the ectoparasites? Is the immune response involved? Shedding?
Yeah, that's, we believe what's happening is the lizards are shedding, and that gets so the ectoparasites are mites. And so the mites work their way into the scales, and so the cost of shedding is too high for the lizards in that warm experimental temperature treatment. The control lizards are in better condition, so I think they're able to slough off their skin, and that lowers the ectoparasite load. So I think that's the case. I also think that the lizards that are in the high nocturnal temperature treatment, they're I think they're immunocompromised, but we didn't measure that, so we don't know. But we do know that there's no difference in oxidative stress. So any of the ROS assays showed no difference.
So our fearless leader, Dr. Jonathan Mossas, has two questions after commenting that you gave such a nice talk. He asks, first, why were the nighttime temperatures higher at one of the sort of high elevation sites? And second, he wants to know how higher nighttime temperatures mechanistically cause some of the changes that you observed.
Good questions. So that one site, Barnasac, it's not super high. It's at an elevation of, I think, 1,500 meters. But the thing about it is it's a bowl, right? So you've got this this rugose topography. So there's these rolling hills, and so this particular site, there's a prairie on one side, and then the lizards are found in this bowl. So all the lizards are concentrated in this thermally protected environment. And so temperatures outside that bowl are cooler. But this thing, it captures heat and then retains it. So that's why it's warmer. We were baffled. We're like, this is the highest elevation. It's supposed to be cold. And it wasn't. So we went back and we took some temperature measurements, and we said, oh, okay, yeah, it's it's warmer. And we noticed that when we were catching lizards, that the temperatures down in the bowl were warmer than when we were climbed up out of it. Now, the mechanistic part of changes, the morphological changes, I think what's happening is, and this is something we were going to do last year, but we didn't have the opportunity. But we have a microrespirometry set up, and what we were going to do is to measure metabolic rate of the yearlings during the day and then at night to see how much that increase in temperature raises their metabolic rate. And so we think it's an energy allocation response. So there's no difference in the amount of food that the lizards are getting. So they're fed the same amount. Everything's the same except the lizards were put into a room that was 4 degrees C higher. So what we're thinking is the increase in length comes at a cost of putting on fat. So whatever residual energy is available for storage, they're burning it because of higher temperatures overnight. And so one thing to do would be to take the lizards and measure resting metabolic rate at different temperatures. So at the control temperature, 2 degrees and 4 degrees, to see what the rate of increase in metabolic rate is, and then ask. You can predict what the energy expenditure is, and then work backwards to see if that explains the difference. The other thing to do would be to use doubly labeled water to get a measurement of whole organismal energy expenditure. So it's not just how much O2 or CO2 that's being produced, but how is that energy being used in terms of the cost of existence, basically. So I'm going to go with metabolic rate. And if Jonathan has another idea, he needs to come to France and test it.
It's always metabolic rate, isn't it? Isn't that always the answer?
It's standard. I have it in my wallet, metabolic rate.
All right. Well, thank you again, Dr. Miles, for such a wonderful and fascinating talk. I think we're going to go ahead and wrap it up there. So I want to, before we go, I just want to thank the audience for sticking with us for this great talk. And we hope to see everyone next week. So.
And to the streaming.